Today, FDA approvals in breast cancer and prostate cancer, a priority review designation in breast cancer, a breakthrough therapy designation in hepatocellular carcinoma, and new drug applications in acute myeloid leukemia and multiple myeloma. Welcome to Enclave News Network. I'm Gina Columbus. The FDA has approved upfront ribociclib for use in combination with an aromatase inhibitor for the treatment of pre- or perimenopausal or postmenopausal women with hormone receptor positive, HER2-negative, advanced or metastatic breast cancer. Ribociclib was also approved for use in combination with fulvestrant for the treatment of postmenopausal women with HR-positive, HER2-negative, advanced or metastatic disease in the frontline setting or after disease progression on endocrine therapy. The decision for the ribociclib aromatase inhibitor regimen was based on the Phase 3 MONALEESA-7 trial, which showed that combining ribociclib with an aromatase inhibitor resulted in a 14-month improvement in median progression-free survival versus an aromatase inhibitor alone. The approval for ribociclib plus fulvestrant was based on data from the Phase 3 MONALEESA-3 trial, in which the median PFS was 20.5 months in patients randomized to the ribociclib combination versus 12.8 months in those randomized to fulvestrant plus placebo. This marks the first approval that the FDA has granted as part of two new pilot programs announced earlier this year, which are real-time oncology review and assessment aid. Under real-time oncology review, the FDA can review pivotal clinical trial data as soon as it becomes available prior to the formal submission of an application. Assessment aid is a new template for submitting an application that is structured to facilitate a streamlined process for the agency to review the application. In prostate cancer, the FDA has approved enzalutamide for the treatment of patients with non-metastatic castration-resistant prostate cancer. The approval is based on findings from the double-blind Phase 3 PROSPER trial, in which the combination of enzalutamide and androgen deprivation therapy reduced the risk of metastases or death by 71% compared with ADT alone for patients with non-metastatic CRPC. In the study, the median metastasis-free survival was 36.6 months with enzalutamide plus ADT versus 14.7 months with ADT alone. Additionally, there was a 93% reduction in the risk of prostate-specific antigen progression in the enzalutamide arm versus ADT alone. The median time to PSA progression in the enzalutamide group was 37.2 months versus 3.9 months for ADT alone. At the time of the analysis, overall survival data were not yet mature. However, there was a trend toward improvement in OS with the addition of enzalutamide. In triple-negative breast cancer, the FDA granted a priority review designation to a biologics license application for the antibody-drug conjugate sacituzumab govitecan for the treatment of patients with metastatic triple-negative breast cancer following at least 2 prior therapies for metastatic disease. Phase 2 findings with sacituzumab govitecan showed an objective response rate of 34% in patients with heavily pretreated metastatic TNBC. In the 110-patient single-arm trial, the ORR was accompanied by stable disease for more than 6 months in 11% of patients for an overall disease control rate of 45%. The median progression-free survival with sacituzumab govitecan was 5.5 months, and the median overall survival was 12.7 months. Under the Prescription Drug User Fee Act, the FDA is scheduled to make its decision on the BLA by January 18, 2019. The FDA has granted the combination of atezolizumab and bevacizumab a breakthrough therapy designation as a first-line treatment for patients with advanced or metastatic hepatocellular carcinoma. The decision is based on a Phase 1b study in which independent reviewers determined that at a median follow-up of 10.3 months, atezolizumab and bevacizumab induced an overall response rate of 65% among 23 evaluable patients. The 65% ORR comprised a complete response rate of 4% and a partial response rate of 61%. Additionally, 7 patients had stable disease and 1 patient had progressive disease. A disease control rate of more than 6 months was reported for 16 patients. The ongoing Phase 3 IMbrave 150 study is comparing atezolizumab and bevacizumab with sorafenib in the frontline setting for patients with locally advanced or metastatic HCC. In acute myeloid leukemia, a supplemental new drug application has been submitted to the FDA for venetoclax in combination with a hypomethylating agent or low-dose cytarabine for the first-line treatment of patients who are ineligible for intensive chemotherapy. The application is based on two phase 1b/2 trials in this setting, which are the M14358 study and the M14387 study. In M14358, combining venetoclax with azacitidine or decitabine led to a complete remission or CR with incomplete blood count recovery rate of 73%. The median overall survival at more than 1 year of follow-up was 17.5 months. The open-label phase 1b/2 dose escalation and expansion M14387 study examined venetoclax in combination with LDAC. The CR/CRI rate with the combination in this study was 62%. The median OS at more than 1 year of follow-up was 11.4 months. The FDA breakthrough designation expedites the development and review of treatments that have demonstrated early potential to improve outcomes over standard therapy for a serious condition. A rolling submission of an FDA new drug application has been submitted for selinexor for the treatment of patients with penta-refractory multiple myeloma. The application is based on data from Part 2 of the Phase 2b STORM trial, in which selinexor induced an overall response rate of 25.4% in patients with penta-refractory disease. The responses included 2 complete responses and 29 partial responses or very good partial responses. The median duration of response was 4.4 months. Prior data from Part 1 of the study showed that selinexor achieved an ORR of 20.5% in 78 patients with quad or penta-refractory myeloma. Karyopharm, the manufacturer of selinexor, reported in a press release that they plan to complete the NDA by the end of 2018. The FDA previously granted selinexor both orphan drug and fast-track status in this setting. Karyopharm intends to present full data from the Phase 2b STORM trial at an upcoming oncology conference. This week, we sat down with Dr. Adi Diab of the University of Texas MD Anderson Cancer Center to discuss immunotherapy progress in oncology and the role of interleukin-2.
I want to talk about the immunotherapy progress over the last 10 years. We definitely beat the skepticism about the efficacy of immunotherapy. And that was a major hurdle. Many people were not believers because they haven't seen the translation of the science into clinical practice. And in the last 8 years, we have seen that translation. And as it's expected in the field of oncology, with the new treatment, there is sometimes a great hopes and sometimes unrealistic hopes that accelerate more than the science accelerate and advance to understand how to overcome the resistance for the current immune therapeutic regimens. So there is definitely hope and there is definitely expectations. Our fear that we may enter quickly a phase of disappointment, premature disappointment from immunotherapy, and that should not be the case. And because we need to understand better the immunotherapy and understand the mechanism of resistance, this is not for one answer for all patients. This has to be tailored. Sometimes has to be tailored for one patient because inside that patient's different lesions of different tumors behave differently, and science has to be advanced. We have to obtain biopsies to understand that. So the hype and the hope are there, and we just need to maintain that balance as we advance and improve immunotherapy. The high-dose IL-2 toxicity depends on largely into 2 mechanisms. One, the rapid and the overactive immune stimulation of the immune system because of the IL-2 immediately activate those 2 cells. And once you have this rapid proliferations, you have chemical reactions that lead to vasodilation and hypotension, and that's associated with less perfusion of blood into the organs. So that lead to organ dysfunction, sometimes lead to organ dysfunctions in the kidneys, in the heart, and the liver. Another part of the toxicity related to the alpha subunit of the IL-2 receptor, which also expressed on endothelial cells, in particular in the pulmonary, in the lung endothelial cells. And when it's get activated by the IL-2, it leads to leaky vascular syndrome, which is associated with pulmonary edema. And that's become a big hurdle and limit the patient's selection to who can be treated in the IL-2. Nectar-214 is a pegylated cytokine that eventually harness the IL-2 signaling pathway and provide safety profile. One, by being pegylated and the slow release of the pegylation, which is a pH dependent, prevent the or avoid the rapid stimulation of the immune system. So the stimulation of the immune system is there, it's sustained, but it does not peak very quickly and go down very quickly. And that's mitigate some of the immune toxicities that we see with the high dose IL-2. Furthermore, the idea that the pegylation structure is located to block or to bias against binding into the alpha subunit of the IL-2 receptor, that also lead to avoiding of the pulmonary edema. And that's in terms of safety. That drug was designed to be more safe and has a better therapeutic window than the high dose IL-2 and allow it to be tested as an outpatient regimen. And that's what the monotherapy trial with Nectar 214 established. In terms of efficacy, the same thing, the biased signaling through the beta and the gamma subunits of the IL-2 receptor allow for expansion of effector T cells as well as NK cells in the tumor microenvironment, but does not allow for the expansions of the T regulatory cells who depend on the signaling through the alpha subunit when it's merged with the beta and the gamma. And that's why there is a preferential expansion of the good anti-cancer cells versus our regulatory cells who we don't need them to be active while we're having cancer. So that's the structure of the drug that gives it a superiority in terms of safety, and theoretically we have a better immune stimulation bias towards effector and less towards inhibitory cells.
That's all for today. Thank you for watching Enclave News Network. I'm Gina Columbus.